Module 4, Chapter 19, the cardiovascular system, the heart. Now that you've become familiar with the blood and all that it does for you, we're going to turn our attention to the heart, the structure that creates the force to move all of that blood around your body. Now the cardiovascular system is composed of two circuits. The pulmonary circuit involves blood flow to and from the exchange surfaces in the lungs. The systemic circuit is blood flow to and from the rest of the body. There are three types of blood vessels that carry out the function of moving blood around the body. Now these three types of vessels are going to be discussed in great detail in Chapter 20. However, there are blood vessels associated with the heart, so you do need to know the basics. Arteries always carry blood away from the heart. Veins always carry blood toward the heart. Capillaries connect the two, so they allow blood to pass from the arteries through that capillary and into a vein. Veins are referred to as your exchange vessels because they allow for the movement of materials between blood and tissues. So let's talk a little bit about the location and orientation of your heart. With regards to size, your heart is approximately the size of a closed fist. It is located in an area of your thoracic cavity called the mediastinum. The mediastinum is best shown in the upper image on the slide, which is showing a cross-sectional view through the thoracic cavity. The mediastinum is located between the two pleural cavities, which house the lungs, and between the sternum and vertebrae. In the mediastinum, you will find your great vessels, which are the largest arteries, the pulmonary trunk and aorta, and the largest veins, the superior and inferior venae cavae. You'll also find in the mediastinum the thymus, which is part of your lymphoid system, the esophagus, which is part of your digestive system, the trachea, which is part of the respiratory system, and your heart, of course. Now, on average, the average adult heart weighs approximately 300 grams. The heart is surrounded by the pericardium. The pericardium isolates the heart from the other organs in the thoracic cavity. It gives the heart room to expand, yet resists excessive expansion that could potentially damage the heart, and helps to hold the heart in its position. The pericardium is composed of two layers. The outermost layer is a layer of dense, irregular connective tissue called the fibrous pericardium. It is equal in strength to the dura mater surrounding the brain. Deep to the fibrous pericardium, you have a two-layer serous membrane called the serous pericardium. The serous pericardium has two components to it. The parietal layer of the serous pericardium lines the inner surface of the fibrous pericardium. The visceral layer of the serous pericardium, which is also called the epicardium, adheres to the surface of the heart. Because it is physically attached to the heart's surface, it is considered part of your heart wall. The space located between the parietal layer and visceral layer is called the pericardial cavity. It is filled with a serous fluid called pericardial fluid that helps to lubricate the parietal layer and visceral layer and allows for the heart to contract without the creation of friction. Two-thirds of the mass of the heart is actually located to the left of midline. The heart has both an apex and a base. The apex is the inferior pointed tip of the heart. It is tipped to the apex is tipped to the anterior, inferior, and to the left of the midline, and can be found at approximately the left fifth intercostal space. The base of the heart is the broad superior part, which has the great vessels attached to it. So you can describe the base as being tipped to the posterior, superior, and to the right of midline. It is found at the level of the third costal cartilage. So now let's talk a little bit about cardiac muscle tissue and sort of spotlight how it's different from the skeletal muscle tissue that you learned about in A&P 1. Cardiac muscle cells are short, thick, branching cells. Recall that skeletal muscle cells, or muscle fibers, were long and straight, sort of like cylinders. The short branching cells of cardiac muscle are connected to each other through specialized junctions called intercalated discs. In the intercalated disc, you can find interdigitating folds. You can see these folds in the lower image on the slide. You have small folds of plasma membrane in one cell and in the neighbor, and the folds fit into each other, sort of like putting your fingers of both hands together and having the fingers of your right hands inserting themselves between the fingers of your left hand. These interdigitating folds form an interlocking mechanism that helps to hold the cells together. You also have some unique cell junctions inside your intercalated discs. You have something called a fascia adherens. Now, this is your most extensive cellular junction. It's a broad band of actin, a type of protein, which is anchored to the plasma membrane and helps to link one cell to the next. You have desmosomes, which are focal spots of plaque on the inside surface of the plasma membrane that connect to similar plaques of protein in the adjacent cells. You learned in Chapter 4 of A&P 1 that desmosomes help to prevent cells from separating under extreme tension, like nice strong cardiac muscle contractions. So you can say that the desmosomes and the fascia adherens are going to help you with physical attachment of your cardiac muscle cells. You also have gap junctions, which are those little teeny tunnels that connect the cytoplasm of one cell to the cytoplasm of the next cell. These gap junctions are going to allow the cells to pass ions very quickly throughout the cardiac muscle tissue. And remember from A&P 1, ions are electrically charged, so that by passing ions from one cell to the next through a gap junction, you are in essence passing an electrical current from one cell directly to the next. Your cardiac muscle cells have a single nucleus. Recall that skeletal muscle fibers were multinucleated. They could have up to 100 or possibly more nuclei per cell. There are some other differences. Your cardiac muscle tissue has a very poorly developed sarcoplasmic reticulum. There are no terminal cisternae. However, it does have larger transverse tubules, much larger mitochondria, and there are no myosatellite cells with cardiac muscle tissue. Remember, myosatellite cells in skeletal muscle were used to help repair the cells after damage. Cardiac muscle tissue does not have those myosatellite cells, meaning if cardiac muscle tissue is damaged, it does not have the ability to repair or replace those damaged cells. Your heart can be broken down into 4 chambers. These 4 chambers can then be separated into atria and ventricles. The 2 atria are the smallest and most superior of the heart chambers. They sit on the base of the heart. Each atrium is a receiving chamber for blood returning to the heart. On the anterior surface of each atrium, you have a little wrinkled pouch called an auricle. The auricle has the ability to expand and hold more blood. So you can think of the auricles as being able to increase the blood holding capacity of your atria. The 2 atria are separated internally by a wall called the interatrial septum. The coronary sulcus is a shallow groove on the external surface of the heart. It completely encircles the heart, so it's going around the entire circumference of the heart, and it marks the boundary between the atria and the inferior ventricles. The foramen ovale is a small hole located in the inner atrial septum within the fetal heart. This hole in the fetus is one of the pulmonary bypass mechanisms. You have the ability to move blood from the right atrium into the left atrium and completely pass the pulmonary circuit. Now, as an adult, you don't want to have your pulmonary circuit bypassed. So, at birth, when the now infant takes its first breath, the foramen ovale closes. And over time, it's going to be transformed into something called the fossa ovalis. The fossa ovalis is a depression in the interatrial septum of the adult heart. And for the most part, all it does is mark where the foramen ovale used to be in the fetus. The inferior 2 chambers of the heart, so these are the chambers that help to form the apex of the heart, are called ventricles. They are much larger and much more muscular than their atrial counterparts. With regards to the ventricles, it's important when you look at the heart superficially that you be able to distinguish the right ventricle from the left ventricle. To make this determination, you want to look for 2 sulci that are extending down from base towards apex. One sulcus is called the anterior interventricular sulcus. So this is a groove running along the anterior surface of the heart. The other is the posterior interventricular sulcus. So it's running down the posterior surface of the heart. These 2 sulci mark where the interventricular septum is located inside the heart. It is the interventricular septum that is the solid wall separating right ventricle from left ventricle. Since you can't see the interventricular septum from the surface, these sulci are a clinician's way of knowing where that wall is, so that if you're doing some sort of surgical procedure on the heart, you don't accidentally cut into the wall. So now let's talk about some structural differences between your right and left ventricles and between your ventricles and the atria. With regards to structure, I want to refer specifically to something called the myocardium. The myocardium is the muscular layer of the heart wall, and this muscular layer changes from chamber to chamber. For example, the myocardium in the atria is extremely thin, so the atria do not have a lot of muscle, which means they do have the ability to contract, but they can't contract with any significant force. However, you don't really need the atria to contract with force. Their primary job is to act as a blood receiving chamber. So they're receiving blood, and their only job after receiving it is to pass that blood into the ventricles, which are located inferiorly to them. Now, in the next module, you'll be learning that most of the blood movement between the atria and the ventricles is passive, meaning it doesn't even require muscle contraction to occur. So, if these chambers fill with blood, and the blood moves primarily without muscular contraction, why would the atria need to have a lot of muscle? They don't. Now, if you compare the atria to your ventricles, the ventricles do have much thicker myocardium. The reason for that is that the ventricles are responsible for creating all the force needed to move blood out of the heart and throughout the body. So they're going to have to be able to create a whole lot more force when they contract compared to the atria. However, if you compare left ventricle myocardium to right ventricle myocardium, the two ventricles are not equal. The left ventricle has much more myocardium than the right ventricle. And the reason for this comes down to those two circuits that I defined at the beginning of this module. Your right ventricle pumps blood through the pulmonary circuit. The left ventricle pumps blood throughout the entire systemic circuit. So your left ventricle has to pump blood a much greater distance. It has to create greater pressure to overcome greater resistance to blood flow throughout that systemic circuit. The right ventricle, to be honest, it's only creating enough pressure to move blood to the lungs and back. And the lungs and the heart are at the exact same level, so gravity isn't even an issue with regards to blood flow as well. So your left ventricle just has more myocardium than anyone else because it has to do the most work. Now, between the chambers and between the ventricles and their associated blood vessels, you do have structures called heart valves. The purpose of the heart valves is to make sure blood flow through the heart is one direction only and backflow is prevented. Now, there are two classes of heart valves. You have atrioventricular valves and semilunar valves. Now, the name atrioventricular tells you where these valves are located. They are positioned between the atrium and its corresponding ventricle. Now, the purpose of the atrioventricular valves is to make sure blood flows only from atrium into ventricle. No blood moves from ventricle back to atrium. So these little AV valves are ensuring one-way flow from atrium to ventricle. Now, anatomically speaking, the atrioventricular valves are built from structures called cusps. So cusps are little leaf-shaped flaps of dense irregular connective tissue. And these cusps, these flaps, are attached to the walls of the ventricles through cord-like structures called chordae tendinii. You can also call them tendinous cords. Now, if you look at the figure on the screen, focus in on either one of the labeled atrioventricular valves. The solid white portion would be the cusps, the leaf-shaped flaps. Extending off of the valve, you can see the white cord-like structures. Those are the chordae tendinii. Now, if you follow those cords down towards the ventricular wall, do you see how the cords are attaching themselves to nipple-like projections coming out of the ventricular wall? Those little nipple-like projections are called papillary muscles. The chordae tendinii only anchor to papillary muscles. Now, the atrioventricular valves are specifically named. The right atrioventricular valve, or you can call it the tricuspid valve, is positioned between the right atrium and the right ventricle. The left atrioventricular valve, or mitral valve, is positioned between the left atrium and the left ventricle. Please note that the word bicuspid valve is not in the lecture notes. The term bicuspid valve does not exist in the International Terminology Standard. The only two acceptable terms for this valve are mitral and left atrioventricular. So please be sure to use one of the accepted terms. You will be given no credit in lab or lecture for the use of bicuspid if asked to name this valve. The other class of heart valves are called semilunar valves. The semilunar valves are positioned between the ventricle and its associated blood vessel. What that means is it's between the ventricle and the vessel that the ventricle pumps blood into as it leaves the heart. Now, the purpose of the semilunar valves is to prevent backflow. It's going to ensure that blood will only move from ventricle into vessel. Blood will never move from vessel into ventricle. Now, the reason why they're called semilunar valves is because of the shape of the cusps. The cusps are actually shaped like little half moons. So because they look like little half moons, they're referred to as semilunar. And both semilunar valves consist of 3 moon-shaped cusps. You're going to love seeing these valves on your sheep hearts in lab. You're actually able to move them with the blunt probe, and you can actually simulate their function and reinforce your lecture notes right there on an actual heart. Now, there are 2 different semilunar valves. The first one is called the pulmonary valve. The pulmonary valve is positioned between your right ventricle and the pulmonary trunk, which is the main artery that begins the voyage of blood flow to the lungs. The aortic valve is positioned between your left ventricle and your aorta. The aorta carries blood throughout the entire systemic circuit. Now, on this figure, the aortic valve is actually posterior to the pulmonary trunk, so you are unable to see it in this particular image. Now, these heart valves are only composed of dense, irregular connective tissue. They do not have any muscle in them, so they don't actually contract and create movement. So you might be wondering, then if they can't contract, they can't move on their own, what causes them to open and close? And the answer to that question is pressure. As your chambers are contracting and relaxing, you are going to have pressure changes within the heart. These pressure changes are going to then be responsible for the opening and closing of the valves. If a chamber is contracting, contraction creates an increase in pressure. Relaxation of a chamber causes a decrease in pressure. And again, these valves and their very precise timing of opening and closing is going to help ensure that blood flow through the heart is one direction, and that inappropriate backflow through the heart will not occur in a healthy person. Now, before I get into the nitty-gritty of how the different valves open and close, and when they open and close, I wanted to give you a summary of it. A little snippet that if you can remember these two statements, you can then figure out everything else that's to come. So with atrioventricular valves, these valves are open when your ventricles are relaxed, and they close when your ventricles contract. Now, if you can remember that one statement, your semilunar valves are always the opposite. Your semilunar valves close when your ventricles relax and open when your ventricles contract. All right, so now let's look a little bit more specifically about the atrioventricular valves and how they close and open. Now, I want you to imagine the image on the left-hand side of the screen. This image is showing you the point in time when your ventricles are completely relaxed. So there's no muscular contraction in the ventricles at all. At this point in time, the atrioventricular valves are open. The points of the cusps are extending down into the ventricle. The papillary muscles are nice and relaxed. Again, no muscular contraction anywhere in the ventricles. And the chordae tendinii are slack, meaning there's no tension on those cords. At this point in time, if you were to compare the pressure inside the atria and inside the ventricles, your atria have a higher pressure than the ventricles at this time. So blood loves to move from an area of high pressure to an area of low pressure. So while these atrioventricular valves are open, you're going to see blood flow from the atria down into the ventricles. Now I want you to imagine that your ventricles change what they're doing, and your ventricles go from being relaxed to the image on the right-hand side of the screen, which is the ventricles contracting. And muscular contraction increases pressure. So your ventricles start to contract, and the increase in pressure that you start to build inside the ventricle starts to actually push blood superiorly in that chamber, as you can see by the yellow arrows on the screen. And what happens is that this blood pushes up on those little atrioventricular valve cusps, and it causes the cusps to move upward, to move superiorly until they meet in the center of the opening. And when all of these little cusps meet, they completely seal off the opening between the ventricle and the atrium, preventing backflow. However, you're going to create a lot of pressure in this little ventricle. And the last thing you want is for the pressure in the ventricle to push on those atrioventricular valves and cause those cups to evert and create an opening into your atrium, which is why as the ventricles are contracting, the papillary muscles are contracting too. So those little nipple-like projections are all increasing their tension. When they contract, they're going to tighten the chordae tendinii. So those little cord-like tendons are going to get very, very tight. And they're going to serve as an anchor that helps prevent your atrioventricular valves from everting. They allow enough movement so that the cusps can lift and meet and close the opening, but they're not allowed to move any further than that because of this muscular bracing. Now, closing the atrioventricular valve is important because it prevents blood from moving from the high pressure in the ventricle to the low pressure that is now in those atria. So it's going to help prevent backflow. Now, let's talk about those semilunar valves. And let's pick up exactly where we left off. We're going to look at the image on the left-hand side. So we've got these ventricles. They are contracting, they are creating pressure, and as we mentioned, that increase in pressure closes the atrioventricular valves. However, you're going to increase the pressure in the ventricles so much that your ventricles become a high and the pressure of blood in their vessels becomes the low. So your left ventricle ends up with a higher pressure than the aorta. The right ventricle gets a higher pressure than the pulmonary trunk. And this pressure surging up from your ventricle actually presses up on the semilunar valves. It presses on those little teeny tiny moon-shaped cusps. And as you can see in the figure, by that large arrow heading up into that blue pulmonary trunk, it's going to push those cusps to the side and actually flatten the cusps against the inside wall of the artery. And by pushing those cups to the side, you actually create a nice large opening that's going to allow blood to move from the ventricle and up into that vessel. Now, your ventricles, after they're done contracting, they will shift back into their relaxation phase, which is now transitioning to the right side of the screen. When your ventricles relax, they become a source of very low pressure. And if you compare your ventricle to the pressure in its vessels, your vessels are now the high and your ventricles are the low. And blood loves to move from high to low. However, you don't want backflow. You do not want blood to flow from those vessels and back into the ventricle. So what's going to happen is as blood attempts to backflow into the ventricle, the blood actually slips behind the cusps of the semilunar valve and causes them to swell. And when those little moon-shaped hammocks swell with blood and expand, they actually meet in the middle of the opening and seal it shut. So when you're dealing with the semilunar valves, there is no need for muscular bracing. There's no chordae tendinii, there's no papillary muscles involved. It's high pressure in the ventricle, flattening the valve cusps to create an opening to move blood out of the heart and into the vessel, and then blood filling the cusps, causing them to expand to close the opening to prevent backflow. Now, things can go wrong with your heart valves. For example, you could have stenosis in any one of those heart valves. Stenosis is a narrowing of the heart valve, and if the opening in the valve is narrower than it should be, it's going to restrict blood flow through it, which is going to impede your ability to move blood from an atrium to a ventricle or from a ventricle into a vessel. Now, stenosis can be due to scarring, so if perhaps you had an infection in the heart, cleared the infection with medication, but that infection caused some heart valve damage. The damage might lead to the development of scar tissue, which would cause the stenosis. You can also be born with it, so it is possible to be born with a valve that is narrower than normal. If you're dealing with mitral valve prolapse, as the name implies, is dealing with the mitral valve. Now, the mitral valve is built out of only two cusps, so it's got two little flaps of tissue. And in mitral valve prolapse, one or both of the cusps actually evert when your ventricles contract. And this eversion is going to allow backflow from your left ventricle into your left atrium. Now, this backflow is actually audible. You could actually hear it with a stethoscope. It would sound like a little swishing sound in between your normal heart sounds. Now, mitral valve prolapse is an example of something called a heart murmur. Heart murmurs are when you do have backflow through a heart valve, and you can get a heart murmur, this inappropriate backflow, in any of the valves. It's just that mitral valve prolapse is the most common type of heart valve because your left ventricle, with all of that muscle, creates the highest pressure inside the heart. And when you have a chamber creating such intense pressure with every cardiac cycle, the valve, that little mitral valve, is taking a beating. So with regards to the heart murmurs, they can occur in any valve. It's just the mitral valve prolapse, because of where it is the most susceptible. You also then need to be concerned about rheumatic fever. Now, rheumatic fever is most common in children, but it can happen in adults. There was a faculty member here at the college about 10 years ago that actually got strep throat and developed rheumatic fever as a secondary issue after the strep throat infection had completely cleared. So it is most common in children, not uncommon in adults. Now, rheumatic fever is caused by streptococcal infections. So if you suspect that you have strep throat, you need to go to the doctor and get it looked at. Untreated strep infections will lead to rheumatic fever. Now, rheumatic fever itself is an autoimmune disease. Now, that term autoimmune means that your lymphoid system, which is sometimes nicknamed the immune system, actually starts to attack normal body tissues. So it develops a glitch where it starts to attack tissues that are supposed to be in the body. And it targets specifically the connective tissues of the body. And your heart valves are all made out of dense, irregular connective tissue. So rheumatic fever, as it starts to attack connective tissues throughout the whole body, it's attacking the connective tissues within the heart as well. So it's going to cause a lot of inflammation within the heart. It can also lead to permanent valve damage as well. In some severe cases, even requiring valve replacement because the valve is so destroyed as a result of the rheumatic fever. Now, at this point, what I'd like to do is sort of take all of the different parts of the heart and tie them together into one big story. As if you are a blood drop, You are coming from some place in the body. You are deoxygenated. So you need to return to the heart so that you can then be pumped through the pulmonary circuit to the lungs where you can pick up oxygen. Return to the heart through the pulmonary circuit, and then leave the heart through this and through the aorta to then flow through the systemic circuit to get that oxygen out to the body. So we're going to pretend we're a little blood drop somewhere. We are in desperate need of O2. Now, I will be honest with you. On the very first exam, there is a 10-point essay question. This one slide is that essay question. You will be asked to describe the flow of a blood drop through the heart. Now, I will tell you where in the body that little drop is coming from. So you're going to have to tell me what blood vessel it is traveling through when it returns to the heart. But you need to be able to trace the voyage. You also will need to be able to indicate, as the blood is passing through every single structure, whether that blood drop is deoxygenated, so oxygen poor, or oxygenated, meaning oxygen rich. I personally recommend using a negative sign next to the term to indicate deoxygenated, and a plus sign next to the term, meaning it is oxygenated. And for the essay question, you can do it however you would like. You can do it as a flowchart, just sort of term with an arrow, and just show me that you understand through the flowchart how that blood drop is moving. You can write it out in paragraph form if that's a format that you feel more comfortable with. You can even do it as the list of A through N that I have in your lecture notes for you. Just add the pluses and minuses to indicate oxygenated and deoxygenated. Now, what I've done is I have numbered the different structures that the blood drop is passing through on the screen. So, as I mentioned the structure in the notes, I'll mention what number I'm on. So, you've got a blood drop. If the blood drop is coming from body structures superior to your diaphragm. Now remember, in A&P1, this is why you had to learn the diaphragm. The diaphragm is that large, sheet-like muscle that separates your thoracic cavity from your abdominopelvic cavity. So if you are a blood drop coming from any body structure superior to that diaphragm, and that would include the upper limbs because they attach to the torso at the shoulder, and the shoulder is superior to your diaphragm. If you are diaphragm and higher, you are going to return to the heart through structure 1A on the figure, your superior vena cava. If you are returning to the heart from a body region inferior to the diaphragm, you are returning to the heart through 1B, your inferior vena cava. Now, if the blood drop is coming from the myocardium of the heart, so if this is blood that has been circulating through the myocardium, supplying oxygen to the cardiac muscle tissue, that blood is going to flow through coronary veins, and it's going to collect in a structure called the coronary sinus. So the coronary sinus receives blood from the myocardium of the heart. So all three of these vessels are deoxygenated, so they all should have minus. Now blood from these three vessels is going to flow into number 2 on the figure, which is your right atrium. Now we have not gotten to the lungs yet, so the blood in the right atrium is still minus, still deoxygenated. From the right atrium, blood is going to flow through number 3, the right atrioventricular valve, so put a little minus next to the term. It then enters your right ventricle, which is number 4. Still have not made it to the lungs, still minus. Your ventricle is going to contract. It's going to push the blood up through number 5, your pulmonary valve, still minus, into a single short artery, number 6, called the pulmonary trunk. Now we still haven't gotten to the lungs, so the pulmonary trunk, minus. Just under the aorta, the pulmonary trunk is going to split. It's going to hit a T intersection. It's going to send a branch to the right and a branch to the left. So it's going to send blood through 7A, which is your right pulmonary artery, and 7B, which is your left pulmonary artery. Now, the pulmonary arteries are going to send blood out into the lungs. The blood is going to end up passing through the pulmonary capillaries. Now, it's a little tricky here because the blood coming into the capillaries is minus, but when it leaves the capillaries, it's plus. So what I would do is I would do minus and then a slash and a plus to indicate that in the lungs we change our oxygenation status from deoxygenated to oxygenated. So this is awesome. We've got your nice deoxygenated blood drop successfully into your pulmonary capillaries. However, your lungs are not connected to your feet or your digestive structures or your brain. So that nice deoxygenated blood drop now has to come back to the heart so that it can head out into the systemic circuit. So we're going to use pulmonary veins. We have got 2 pulmonary veins on the right, which is the 2 for 8A, and 2 pulmonary veins that are left pulmonary veins that are 8B. Now, the pulmonary veins are bringing the blood from the lungs. They're bringing that blood back to the heart and deliver their blood into number 9, which is the left atrium. Now I want you to think about this. The pulmonary veins are carrying oxygenated blood out of the lungs. So the pulmonary veins would be plus. Likewise, the blood in the left atrium would also be plus. The blood flows from the left atrium through number 10, which is your left atrioventricular valve, still plus, into number 11, which is your left ventricle, still plus. Your left ventricle is going to contract. It's going to push the blood up through the aortic valve, which you cannot see on this figure. If you could, it would be number 12. It's actually behind number 6, your pulmonary trunk. Through the aortic valve, which would be plus, into your aorta, which is number 13. The aorta, specifically the short segment coming up out of the heart, which is your ascending aorta, is positive. It's fully oxygenated, and it now can send blood out to your systemic capillaries to supply your tissues with oxygen. So now we're going to wrap up this module with the beginning of our cardiac physiology. When you start thinking about your heart and its ability to contract, it brings to mind that the tissue of the heart contains 2 different types of cells. You have cells of a conducting system, which are responsible for controlling and coordinating your heartbeat. You also then have contractile cells, which are responsible for producing powerful contractions that propel blood through your pulmonary and systemic circuits. Now, when you start thinking about this coordinated heartbeat, you need to think about the term cardiac cycle. The cardiac cycle is one complete contraction and relaxation of all four heart chambers. So basically, it's the period of time between the start of one heartbeat and the start of the next. Your cardiac cycle is going to begin with a small little structure we're going to talk about a little bit more about, called the sinoatrial node. And this one little node produces the action potential that actually begins each heartbeat. So this action potential has the ability to pass through the entire myocardium of the heart and stimulate the contraction of your contractile cells. Now, what we're going to do to wrap up this module is we're going to talk about the conducting system. So we're going to talk about these specialized cells that form it. Now, your heart possesses specialized cardiac muscle cells called autorhythmic cells. They only make up about 1% of all the cells in the heart. These specialized cells, if you think about their name, autorhythmic, it means that they are self-excitable. They will automatically excite themselves. And they will excite themselves in a rhythm. So they will repeatedly generate the action potentials that trigger your heart contraction. Now, this creates something very unique because there is no need for neural or hormonal stimulation. These cells will stimulate themselves to produce action potentials and drive your cardiac cycles without input from the brain, spinal cord, or a gland. So for those science fiction movies, when they rip the heart out of someone's chest and they're holding it in their hand and the heart is still contracting, that's not biologically inaccurate. Your heart will continue to contract on its own outside the body as long as the cardiac muscle tissue is well supplied with nutrients and oxygen. It's only when those things run out and you can no longer produce ATP that the heart itself will die. So the next time you see that in a movie, you can nerd out and tell everyone that you're sitting with that it's biologically accurate. Now, these specialized cells are going to kind of act like a pacemaker for the heart. So they're going to keep that pace, that regular rhythm. And they're very highly organized. And they're going to establish something called a conduction system within the heart. This conduction system is a network of these specialized cardiac muscle cells, and this network provides a path for each cycle of cardiac excitation to progress through the heart. So you basically have internal wiring, and in our heart lab, we actually do have a conduction system component because we in the Venice campus are privileged to have conduction system models where you actually can see this wiring inside the heart and really clearly visualize how those action potentials are moving. So you want to use that section of the lab manual to really enhance your understanding of this component in the lecture notes. Now, this conduction system and this organization is significant because it's going to ensure that all of the heart chambers are stimulated to contract in a coordinated manner. And with regards to coordination, when you start your cardiac cycle, it is the atria that will contract first, followed by the ventricles. So it's always atria, then ventricle, atria, then ventricle. And that pacing is established by this basically electrical wiring. Now, when you start thinking about the conduction system and its components, we're going to start by talking about the sinoatrial node, which is abbreviated SA node. Now, the sinoatrial node is located in the right atrium near the opening to the superior vena cava. So it's in the right atrium wall near the superior vena cava. And this little node possesses what's called a pacemaker potential. And what that means is that the cells in this node cannot maintain a stable resting membrane potential, meaning they will automatically and gradually depolarize to threshold after each repolarization. So remember, repolarization was your return to rest. Well, these little cells, the moment that you return to your resting membrane potential, they automatically start to depolarize back to threshold to begin another action potential. So these cells never stop. They're always undergoing an action potential. Now, they're going to establish something called a sinus rhythm. This is an individual's normal resting heart rate. Okay, they're going to establish this sinus rhythm. And because this structure is the structure to create that action potential at the beginning of every cycle, it's sometimes referred to as the pacemaker. Now, if left to its own devices, the sinoatrial node will actually create approximately 100 action potentials per minute. So that little node all on its own will create 100 action potentials per minute. However, a healthy person does not have a resting heart rate that is 100 beats per minute. Average resting heart rate is usually in the 60s or 70s. It's a little bit lower. This lower resting heart rate is due to the fact that the parasympathetic division of the autonomic nervous system can slow the sinoatrial node down. So the sinoatrial node will create action potentials without the brain's input, without the spinal cord's input, without a hormone's input. But you can influence it. You can slow it down. We're also going to talk about how you can speed it up. Now, the sinoatrial node, so imagine it has sent off this giant burst of electricity. The action potential from here is now going to spread throughout both of the atria through what are called conducting cells. Now, you have a structure called you have a interconnecting network in the atria called the internodal pathway. And the internodal pathway is designed to carry the action potential from the sinoatrial node through the wall of your right atrium to your next node, which is called the atrioventricular node. However, if you think about that, it implies that the internodal pathway only carries the action potential throughout the muscle of the right atrium, which means that you do have to have a component of the internodal pathway that extends across the interatrial septum into your left atrium. And that little structure is called the interatrial bundle. It's specialized cells that will take the action potential made by the sinoatrial node past that electrical signal through the interatrial septum and then throughout the muscle of the left atrium. Now, as you are passing the action potential through the internodal pathway and through the interatrial bundle, you are stimulating the muscle of each atrium to contract. So the movement of the action potential through the atria is going to stimulate atrial contraction. All right, so now all we have done is create an action potential. We have passed it through the atrial walls, causing your atria to contract, and we deposit that action potential at a little node, which is number 3 in the figure on the screen. It is called your atrioventricular node, or your AV node. This node is located in the interatrial septum. It also does possess a pacemaker potential. However, it only beats about 40 to 50, it will only create about 40 to 50 action potentials per minute. So it usually is overridden by the sinoatrial node. The SA node fires first, it fires faster, so the atrioventricular node basically keeps pace with the SA node. However, if the sinoatrial node fails, the atrioventricular node can create enough action potentials per minute to keep you alive. So think of this as a backup node. And the rhythm that the AV node would create on its own is called a nodal rhythm. Now again, you will not see the nodal rhythm if the SA node is functional. Okay? If that SA node is functional, you have a sinus rhythm. The nodal rhythm is when the AV node is in control. Now, the atrioventricular node is going to create an action potential, and it is going to pass that action potential into another group of conducting cells called the atrioventricular bundle. Now, the atrioventricular bundle in the figure on the screen is the structure, the yellow structure running along the white arrow just below the pulmonary semilunar valve. It's a short, single electrical wire. And it's an important electrical wire because your atrioventricular bundle is the only electrical connection between your atria and your ventricles. If this bundle is ever severed, you will never be able to pass an action potential to the muscle of your ventricles naturally. So basically you could die if this structure is ever damaged. You'll never be able to eject blood from the heart again unless they can hook up a portable device that can stimulate the muscle for you. Now, the atrioventricular bundle approaches your superior end of the interventricular septum. And as you can see, it's going to give rise to multiple branches that are going to extend inferiorly down along the interventricular septum. These are called your bundle branches. And these bundle branches extend all the way down to your apex. Okay, so these bundle branches are number 4. And when they get to the apex, do you see how the yellow conducting structures then turn and actually begin to extend along the outside wall of the heart from apex towards base? These structures extending from apex towards base, number 5 on the figure, are the subendocardiac conducting network. These wires are going to be stimulating the muscle of the ventricles to contract. So the subendocardiac conducting network is going to cause you to contract the muscle of the ventricles, meaning you're going to be able to eject blood out of the heart. Now what's interesting is directionality. If the bundle branches turn and the subendocardiac conducting network starts at the apex, wouldn't the apex get the action potential first? And I'm sure you're thinking yes. But then wouldn't it mean that the muscle in the apex contracts first. Yes. If you watch that subendocardiac conducting network, it's not just passing the action potential from apex towards base, it's causing the muscle itself to contract from apex towards base. So you have directionality to the contraction. And the reason that you have that is look at where your semilunar valves are located. They're located in the base. Your ventricles have to be able to push blood from the apex superiorly towards the base to eject it from the heart. So how the wiring is organized is accommodating the direction that contraction has to occur so that blood is moving appropriately throughout the heart. So, in summary, when you create the action potential, it's going to form first at the sinoatrial node. You're going to pass that action potential through the inner atrial bundle into the left atrium and through the internodal pathway throughout the right atrium, causing your atria to contract. The signal is then passed off to the atrioventricular node. The action potential travels along the atrioventricular bundle, down the bundle branches in the interventricular septum, out through the outer walls of your ventricles from apex towards base, causing your ventricles to contract. Now, I want to stress before I move to the next slide that heartbeat is intrinsic to the heart, meaning that this conduction system, all on its own, will maintain your repetitive cardiac cycles. No nervous system input needed, no endocrine system input needed. However, heart rate, which is the speed of your cardiac cycles, that can be modified. And it's modified through the actions of your autonomic nervous system. It's also modified through the actions of various hormones. And you'll learn about how this works in the last module, Module 6, on this chapter. Now, occasionally problems do occur with pacemaker function. For example, your patient could be tachycardic. Tachycardia is clinically identified when you have over 100 beats per minute. So your sinoatrial node is forming more than 100 action potentials per minute. You could have the opposite, where your heartbeat is a lot lower. So if your heart rate is below 60 beats per minute, you are bradycardic. Now, bradycardia in someone who is endurance trained, so a marathon runner, a cyclist, a long distance swimmer, and someone who has undergone endurance training, bradycardia actually makes the heart more efficient, so it can move the same volume of blood by contracting less. So it actually is a huge energy saver. However, bradycardia in someone who is not endurance trained can actually impair oxygen delivery throughout the body because your blood is not flowing as quickly as it needs to be. You also could have an ectopic focus. An ectopic focus is any region other than the sinoatrial node that spontaneously starts to fire and produce action potentials. Now, this ectopic focus is usually faster than the sinoatrial node, meaning it can completely override it, and completely or partially override the functions of the entire conduction system. So depending on where the ectopic focus is, you could have some really severe impairment with regards to blood flow through your heart. So always make sure you get that checked out.